Welcome to the 7th season of the South Haven Speakers Series. I'm Mark Odland, and I serve as president of the series. Today's presentation is different in that we are meeting virtually using Zoom webinar technology, but the focus of the series remains the same. The speaker series has never strayed from our mission of addressing issues, innovations, and ideas shaping our lives. Tonight's program by Dr. Clifford Lane addresses a topic on everyone's mind and also goes deep into all three areas of our focus. The issue is the virus. What is it? Where did it come from? How did it get here? How did it spread so quickly? And why did it take so many lives? The innovation part of this is the vaccine and how it was developed so quickly and is so effective. And the idea is how we can work together to defeat and end this pandemic. Issues, innovations, and ideas shaping our lives. Before we introduce Dr. Lane, I want to thank our event sponsors, including Bronson South Haven Hospital, Spectrum Lakeland Health, the Mendel Center at Lake Michigan College, the Kiwanis of South Haven, and the Rotary of South Haven. Thank you for making today's presentation possible. Now, as we get into the program today, if you have questions, you'll see a Q&A box where you can submit those questions, and we will have a Q&A period following Dr. Lane's presentation. I will now turn the program over to our charter board member, Dick Brunvand, who arranged to have Dr. Lane with us today. Dick?
It's February 13th, 2020. You're in Washington, D.C. airport, and you're ready to catch your flight to Japan, where you will help launch a study of an experimental drug which could help in the fight against coronavirus, COVID-19. Then you hear the familiar sound of an email on your phone. Flight plan changed. You're going to China. Dr. Clifford Lane, our speaker, got that email. Never been to China. He probably thought that this may not be the most opportune time for that visit, but as a deputy director of the National Institute of Allergy and Infectious Diseases, who works closely with Dr. Anthony Fauci, the head of NIAID, he was well prepared for his new assignment. Dr. Lane would be one of 2 U.S. scientists to join the World Health Organization's team of 13 international researchers. They would meet up with 12 Chinese colleagues, tour 5 different cities, and then give a first-hand report on what would become a worldwide epidemic. Dr. Lane is a highly credentialed clinician who has been on the front line of HIV and AIDS research, led studies of vaccines and treatments in Liberia during the West African Ebola epidemic. This is just the tip of his many accomplishments, but it's understandable that he's done all this so well because Dr. Lane is a Michigan native, came from Detroit, a 1976 graduate of the U of M, and did his internship and residency at University of Michigan Hospital, then came to the National Institutes of Health in 1979. Now, I'm told that he's an avid Wolverine. That may be an understatement. I'm also told that he hasn't been able to use his tickets that he has for some football games because he's been a little busy. But I also understand that he has taken annual bus trips back to the big house with many of his friends, and just hopefully we'll get things back to normal, and maybe this fall he'll be able to use some of those tickets with his friends. Well, ladies and gentlemen, we welcome from his office in Washington, D.C., Dr. Clifford Lane, Deputy Director of Clinical Research and Special Projects, NIAID. Dr. Lane, we are very pleased to have you here and to kick off our 2021 speaker series in South Haven, Michigan.
Great. Well, thank you very much, Dick. It's a real pleasure for me to be able to share some comments with you and your group. I'm just going to grab the slides. Hope everyone can see that okay. So what I'd like to do this afternoon, this evening, is talk a little bit about some of the work that we've been doing and some of the things that have been happening with regard to COVID-19. We first were made aware of what is now well established as a new outbreak back in January of 2020. It's hard to believe it was only a year ago when reports were emerging from China about novel cases of pneumonia. Within a very short period of time, they identified the cause of this pneumonia as being a novel coronavirus, the same type of virus that causes SARS or MERS. It's also the same type of virus that causes the common cold. And there's some speculation as to whether or not when the common colds first emerged on the scene, if they might not have caused something similar to what we're seeing now with COVID-19. Just in terms of terminology, COVID-19 is the name of the disease that's caused by the virus SARS-CoV-2. And you can see here two different types of electron micrographs of the virus. On the right, a scanning electron micrograph to give you the three-dimensional idea of how this virus is a sphere. And on the left, a cross-sectional electron micrograph where you can see the core, the red core, and then the corona, the halo around the virus in blue that gives it its name. The US, I think, first got pulled into this really back in January of last year due to the first case that was diagnosed in Washington State and a recent traveler to Wuhan, China. At that same time, the Diamond Princess cruise ship departed Yokohama, Japan for a 14-day cruise that was cut short when a patient on board who disembarked in Hong Kong was diagnosed with what's now known as COVID-19. That ship was quarantined in Yokohama. It included a number of US citizens and gave us some of the first detailed epidemiology outside of China, noting that among the 3,711 passengers and crew, 19% became infected. Of those, half had no initial symptoms, and 18% never had a symptom, and 2% of those who became infected aboard the cruise ship died. The US efforts followed shortly behind that. The Chinese were actually very gracious in publishing the sequences of the virus. We worked quickly within the US to identify samples from US survivors, and the Vaccine Research Center of our institute, with a number of scientists, among them Barney Graham, Julie Ledgerwood, John Mascola, took that sequence data to help develop one of the RNA vaccines, the one made by Moderna, and one of the monoclonal antibodies currently being used in treatment made by Lilly. We also started a randomized controlled trial of a repurposed antiviral drug known as remdesivir that had been used, although unsuccessfully, to attempt to treat patients with Ebola virus disease. And as was mentioned, I had the opportunity to be part of a WHO mission to China. I got the word to go on the mission as I was headed to Japan to help set up the remdesivir trial. And here you can see me with a group on the left of a group of Japanese scientists at their center. And then over on the right with my colleague, Dr. Hiromi Imamichi, standing outside the Chinese embassy at night, waiting for someone to come out and take my passport so I could get a visa to travel on to China. That was quite an interesting trip. It arrived on February the 16th, left on the 24th. It involved 25 experts from around the world. It was sponsored by WHO. Bruce Allward of Canada actually headed the mission on the WHO side, and Dr. Liang on the Chinese side. Our report from that mission is published online. For those who have more of an interest in seeing the details, I would encourage attention be paid to the middle part, which is where the data are. Sometimes the descriptions at the beginning or the end do have an element of a spin to them, but I think the data inside is really quite good. If you look at what happened in China, the first cases were diagnosed in early December 2019. Some, but not all of them, were linked to the market in Wuhan, suggesting that there probably were unrecognized cases occurring prior to December. As I noted, the Chinese rapidly identified the cause of these pneumonias as a novel beta coronavirus. Within a matter of weeks, they had developed a diagnostic test. And in a very short period of time, they had seen 75,465 confirmed cases. At its peak, one fever clinic in Wuhan was seeing 500 patients per day. When we think of the scope of where we are now with COVID-19, this seems like a little, but at that time for the Chinese, it was really quite a lot. Indeed, I can remember vividly when the first reports were coming out in January. I actually was on a trip to Liberia and seeing on CNN the fact that 2 new hospitals were being made in Wuhan. This was the Chinese having had experience with SARS, knowing how quickly something like this can become a major public health problem and disruptive. So just as a few notes from the trip to China, there were all sorts of measures put in place and enforced, among them social distancing. Here you can see us having lunch. This was about the only time we were allowed to take our masks off. There was public awareness of the status of the epidemic everywhere you went. While I cannot translate this sign, it gives this community, and this was in one of the residential communities, it gives an update of how many cases had occurred that day in the country, how many in that community, how many people were under quarantine at that point, and a reminder of the things to do to prevent spread. And you can see everyone in the picture wearing a mask. Most of our visits, we traveled by bus, and every so often the bus would be stopped. Someone in personal protective equipment would come on the bus and check everyone's temperatures. While temperature checks may not be the very best way to control an outbreak, it certainly does create an optic or an image that something serious is going on and people need to pay attention. This is a summary of the outbreak in the city of Shenzhen, China. This outbreak for them lasted about 2 months, from January to the middle of February. They rapidly put in place a number of measures to get the infection under control. The first case they actually diagnosed prospectively was here in the middle of January. Even before that time, they had put emergency response measures in place. They had a headquarters for epidemic prevention and control. They closed schools within a matter of weeks, and they came up with guidelines for what needed to be done by everybody. And you can see with these public health measures, no treatments, no vaccines, that outbreak quickly became under control. This looks at the outbreak within China in the green bars. This one peak is when the reporting criteria changed to include non-hospitalized individuals who were infected. And you can see the outbreak beginning here in December, peaking in February, and then really coming under control by March. As it's coming under control in China, you can see it beginning to take hold in other places, in particular Europe. So keep this image in mind, because I'm going to segue now into the global situation and into the US. That picture I just showed you is within this circle. That is the outbreak in China. This is now the outbreak across the world. You can see how heavily hit the Americas and Europe have been, with a total of close to 50 million cases in the Americas, 37 million within Europe. What I'd like to do in the remainder of the talk is touch on several key elements of COVID-19, the epidemiology, the virology, transmission, diagnostics, clinical course, therapeutics, and vaccines. I won't go into great detail, but hopefully we'll have time at the end to answer questions of those areas of greatest concern. This shows the kinetics of the outbreak in the US and the 3 surges that we've had thus far. The one from early spring, the one midsummer, and then the one we're just coming out of from the late fall and going into the winter. We're fortunately on a downward slope at the moment. Everyone is hopeful that will continue. As I'll note in a moment, some of the new variants that we're hearing about gives us reason to worry that that curve may start going up again as some of these new variants, which indeed may be more transmissible, take a greater hold in the US. But again, this is a typical type of pattern that you see with an outbreak for a respiratory virus for which there's little to no pre-existing immunity within the population. At the moment, COVID-19 is the #1 cause of death in the US. If you look at average daily deaths from COVID-19 in the month of January, and then compare that to leading causes of death on a daily basis from 2020, you see 3,000 or so from COVID-19. Fortunately, that number now has dropped pretty dramatically as the overall number of cases has dropped, but followed by heart disease, cancer, Alzheimer's and dementia, stroke, lower respiratory illnesses, a major public health impact. If you compare the total number of deaths from COVID-19, this is as of February 19th, 463,000. You can see, and you often hear this soundbite, it's less than what was seen in the US in the 1918 influenza pandemic, but more than all of the deaths in World War II. Not everyone is at the same risk of death or severe disease from COVID-19. And indeed, we see differences based on race or ethnicity, as well as seeing differences based upon age. You can see the hardest hit in terms of deaths per 100,000 are Black or African Americans, followed by American Indian or Alaska Native. Again, these are rates. Hispanic or Latino, Native Hawaiian and Pacific Islander, followed by white, other, and Asian. Looking at the virus, this is a beta coronavirus. It is the same subgenus as SARS-CoV-1, the cause of SARS, and MERS. It is closely related to some bat coronaviruses, and a lot of the speculation is that SARS-CoV-2 went from a bat to another animal yet to be identified, and from there to human. It is an RNA virus. It has an envelope, as can be seen in the cartoon. In other words, it buds off of cells. It's a positive sense RNA, single strand. It's a very large virus for an RNA virus with a genome of 30,000. It has 4 main structural proteins, the spike protein being the one to which most of the countermeasures, the therapies, the monoclonal antibody therapies, and the vaccines are directed. It binds to cells by binding to ACE2, which is a very heavily expressed receptor on respiratory cells. This shows you on a phylogenetic tree where SARS-CoV-2 is compared to SARS-CoV-1, to MERS, and then these other 4 coronaviruses in red, which are causes of the common cold. One of the biggest worries at the moment is, as we're developing vaccines and antibody countermeasures and other therapies, that we will see variants. And we don't really call them mutants because the virus is constantly evolving. This is the natural course of evolution for a virus. Fortunately, this RNA virus, unlike some other viruses like HIV, has enzymes with it that help make sure that the replication is fairly faithful to the original genome. Despite that, it still does have the ability to drift, to bring in new bases, to bring in new amino acids, such that new variants have emerged. Some of these are called variants of concern because they begin to be the major variant within a population. And depending upon the characteristics of those variants, may indeed have a big impact on where this outbreak is headed. So the first one that became known globally is often referred to as the UK variant, United Kingdom variant. One tries not to necessarily associate variants with locations because it's sort of embarrassing for the location, but it does provide some clarity. This virus is referred to as B117. It does appear to be associated with increased transmissibility. There may be some increased disease severity, but fortunately, there's very little impact on the vaccines or the monoclonal antibodies being used. In contrast, the variant that emerged in South Africa, B1.351, does also have increased transmissibility. It's not entirely clear about its severity. However, what is clear is that at least some of the vaccines being tested and some of the monoclonal antibodies being used may not be as effective against this variant as they are against the wild type or against the B117. In Brazil, the P1 variant of concern has emerged. It looks a bit more like the South African variant than the UK variant, and studies are underway to characterize that virus more fully. To say a few words about transmission, again, this is a respiratory infection. It is mainly spread through exposure to respiratory droplets. Those have their highest concentrations within about 6 feet of an infected person. There are at times aerosols that may contain virus at a distance more than 6 feet. Again, I think you often hear people make very declarative statements about what does not lead to transmission. It really, there are gray zones, and it's hard to live in a gray zone. It's hard to discuss a gray zone. I think the best thing to say is the majority of transmissions probably occur within that 6 foot window. It appears not to be very commonly transmitted through contaminated surfaces, although obviously it's important to keep those surfaces clean, including hands. The virus can be found in a variety of body fluids, but its role in transmission through fluids other than respiratory infections is respiratory secretions, it's fairly unclear. One thing that wasn't initially appreciated, but is fully appreciated now, is that people can be infected with this virus and be completely asymptomatic. Again, different studies will give you different numbers. Somewhere between 1/3 and 1/2 of those individuals infected with SARS-CoV-2 may not show any symptoms at all. And indeed, about 3/4 of the individuals who eventually are diagnosed as having infection may have been asymptomatic at some time. Hence, the notion that you can't tell whether or not someone is infectious just by looking at them, and leading to the recommendations for universal masking at this time. Somewhere around 1/2 of all the transmissions are felt to occur from individuals who indeed are asymptomatic. Some of these are pre-symptomatic. In other words, they're going to develop symptoms. And again, about 1/4 of them probably never develop symptoms. There's a lot of discussion about masks. I won't want to go into any great detail. It's hard to do clear experiments to define what makes the most sense, but I just, more from common sense than anything else, no masks, high risk of transmission. A mask on the infected person will decrease that transmission substantially. A mask on the uninfected person decreases it some, but the mask, this type of mask, the usual surgical mask or the cloth mask, is not good at protecting an individual from getting infected who knows they're going to be exposed. You need to go more with the N95 masks as we use in the hospital setting. You decrease the risk of transmission by the infected person and the uninfected both wearing a mask, and decrease it even more with distancing. So again, some of the fundamental public health measures: universal wearing of masks, physical distancing, avoiding crowds, outdoor contact is better than indoor contact, and frequent hand washing. There are a variety of tests now to make a diagnosis of SARS-CoV-2, namely infection with the virus. The most common test measures directly through amplification the RNA of the virus. You hear this referred to usually as a PCR test. In addition to measure, and these are the most sensitive and the most specific tests, in addition to the test for RNA, one can actually make direct measurements of the proteins of the virus, referred to as antigen tests. Some of the tests that are being put forward for home use utilize this technology. Both of these then directly detect the virus. The third type of test you hear about are the antibody tests. These measure antibodies to the virus. In other words, when an individual becomes infected, they control that infection by making an immune response. When part of that immune response is generating antibodies that bind to and neutralize the virus, and one can measure antibodies in the blood of individuals who have been infected for a period of time, probably 7 to 10 days, at least long enough to make antibodies. This is not helpful in diagnosing acute infection with SARS-CoV-2, but it is of help in diagnosing whether or not someone has ever been infected. One note, though, is that once you get vaccinated, you will make antibodies to the part of the virus in the vaccine, and that's typically the spike protein. The company Ellume has had a big government contract awarded to it to be able to provide antigen testing that could be used at home. I don't honestly know what the timeline is on that, but I think it's something everyone is hoping they'll be able to have in the not too distant future. Regarding the clinical course of illness, this is a very interesting virus that causes an amazing array of clinical illnesses, ranging from no symptoms. I don't know if this picture is from Ann Arbor or not, but it certainly is a bit reminiscent of some of my college days, going from the spectrum of illness from asymptomatic to actually requiring mechanical ventilation and obviously death. The middle shows you a typical chest x-ray from someone with pneumonia due to SARS-CoV-2, a patient with COVID-19 pneumonia. You can see these infiltrates. They're very discrete. They occur in multiple parts of the lung. We often will stage the illness by no symptoms, ambulatory and outpatient with mild symptoms, hospitalized but not requiring oxygen, hospitalized requiring low flow oxygen, high flow oxygen, or mechanical ventilation. Therapeutic strategies are somewhat different depending upon the stage of the illness. Early on, the antiviral strategies probably will have their biggest effect. Later on, it's the immune response to the virus, the inflammatory response to the virus that we think is causing a lot of the end organ damage. So for patients with advanced disease, we look to immunomodulatory strategies. And I'll talk about that in a moment, the steroid dexamethasone being commonly used here. In addition, throughout the course of illness, it appears that SARS-CoV-2 infection leads to a state of increased coagulation, and there's quite a bit of research right now looking at anticoagulation strategies as part of the overall therapeutic management of patients. For people who are infected and have symptoms, about 80% of them have mild disease. And again, this may be only half of the people who are infected, but the ones who are infected with symptoms, about 80% have mild or moderate disease, 14% severe enough disease to be hospitalized, and then 5% with critical disease. The overall case fatality rate is probably somewhere around 2% for this cohort. And if you figure this may be just about half the number of patients infected, the overall case fatality rate for this infection probably around 1%. In addition to pneumonia, there are a variety of other organ systems that may be affected in the setting of SARS-CoV-2 infection or COVID-19 disease, neurologic disorders, cardiac dysfunction, kidney injury, and a rare syndrome in children called multisystem inflammatory syndrome, or MISC, similar to a disease in children called Kawasaki's disease. If you look at the cumulative rates of laboratory confirmed COVID-19 hospitalizations by age, you can see it's really individuals above 50 who make up the bulk of the hospitalization. So the severe disease, more severe disease due to COVID-19, with individuals above 75 really representing the vast majority of individuals who are hospitalized. There are certain underlying medical conditions that make one at increased risk for severe COVID-19 illness. Among them, cancer, particularly in the setting of chemotherapy, chronic kidney disease, chronic lung disease, diabetes, heart conditions, organ transplants, and the associated anti-rejection medicines. Obesity appears to be a significant risk factor, pregnancy, sickle cell disease, and smoking. And we learn more about this all the time. In addition, it's been reported that there's a post-COVID-19 syndrome, or something people are often referring to as long haulers. This is yet to be well delineated, whether it's something unique to COVID-19 or just one of the things that happens to people after a serious illness. Studies are underway comparing people recovering from COVID-19 to those recovering from other illnesses and to control groups to try to delineate what this is clinically, as well as what maybe its cause, symptoms ranging from brain fog to heart damage. I think that at times this may get overplayed a bit, but it's clearly something that's under intense study at the moment. In terms of therapeutics, we don't have a lot to do right now to treat patients with COVID-19. There is one licensed therapy, an antiviral drug called remdesivir. There are a variety of therapies available under something called emergency use authorization. If you look at drugs and the way the Food and Drug Administration, the FDA, regulates drugs, they have a variety of ways to make drugs available to the public. In the setting of a public health declared emergency, which is the setting we are in now, there's a special authority given the FDA called emergency use authorization. Therapies can be made available to the general public under an emergency use authorization if there is some reason to believe that they might be of benefit and there are no good alternatives. That is a lower bar than full FDA licensure, and I think that's an important distinction for people to be aware. Among the things available for treatment under emergency use authorization are convalescent plasma for inpatients, monoclonal antibodies for outpatients at risk, drugs provided in the monoclonal arena from Lilly and Regeneron. A drug called baricitinib is an immunosuppressive agent that's used to treat certain autoimmune diseases. In combination with remdesivir, it has been found to shorten the time to recovery. And then something that didn't require a license because it's already a licensed drug, in fact, off-label, in other words, it's available as a generic, is a drug dexamethasone, a corticosteroid found to markedly enhance survival in patients who have severe disease as defined by a requirement for oxygen. So again, in terms of standard therapy, dexamethasone is probably one of the best tools that we currently have. There are over 2,000 treatment studies underway now trying to find better treatments for COVID-19. Unfortunately, as I mentioned, most of these have not been successful in trying to help the treating community as well as patient community as well as the general community get a better understanding of this. There are a set of NIH treatment guidelines available at COVID-19 treatment guidelines.nih.gov. They review the data available and make recommendations for treatment. Thus far, the one clear recommendation is the one I mentioned initially, the use of dexamethasone for hospitalized patients requiring mechanical ventilation or requiring oxygen, the drug remdesivir for patients a bit earlier in the course of their illness. And right now, the monoclonal antibodies, it's not entirely clear how strong the data are supporting their use, but I think they're increasingly being used. And so again, this is a setting, however, where dexamethasone should not be used because it is a steroid, it is an immunosuppressive drug. And if you use it before the patient has a chance to make their own immune response, you compromise their ability and in fact may make the overall situation worse. These guidelines were requested to be stood up March the 20th last year. The first release was April 7th. There have been 7 major revisions since then and a very large number of page views. So I want to finish up then by making some comments about vaccines. This has been, I think, one of the great success stories thus far for COVID-19 and SARS-CoV-2. It was identified by Science Magazine as the science breakthrough of the year for 2020. There are 3 main different platforms that are being developed and are currently in use. These were heavily supported by the US government by something in the prior administration was referred to as Operation Warp Speed. Many of these same systems are still in play with the new administration. It will probably come up with some other name. I don't think there's another name for it at the moment. So again, there are 3 major platforms that are used: the RNA-based vaccines, the viral vector-based vaccines, and the recombinant protein vaccines. So Moderna and Pfizer, BioNTech are the 2 companies making the messenger RNA vaccines. These can be made, very rapidly. It is a new platform. There are no licensed vaccines with this platform. That we'll come back to in a moment. These are the 2 vaccines that were first out there in emergency use authorization in the US. In fact, are the only 2 out there at the moment. The next platform is the adenovirus vector. AstraZeneca, working with Oxford University, uses a chimpanzee adenovirus as its background. Janssen, part of Johnson & Johnson, uses a human adenovirus 26 as its backbone. These are recombinant viruses, but they are not replication competent. In other words, you get injected with this, the virus gets into the cell, the genetic machinery of the virus has been altered to make the viral spike protein, but that does not then lead to new viruses. So it just presents the spike protein and the other proteins of the virus to the immune system, but does not itself set up an infection. Again, there are no licensed vaccines using these platforms yet either. The final platform, the proteins, recombinant protein, this is standard technology for vaccines, taking again the protein of the spike that has been stabilized by some of the work by Barney Graham and his colleagues at our Vaccine Research Center, and just really injects that protein along with some sort of chemical molecule or adjuvant to help make a better immune response. All of these platforms utilize the spike protein as the antigen. All of the vaccines that are currently in development are directed towards the spike protein. So when we see variants come out, the variants often are variant in the spike protein. So the vaccines may be in a situation where there's going to need to be a constant evolution of what spike is in the different platforms, much the same way that we change the influenza virus vaccine each year. So coming back to where we are with these vaccines, the 2 nucleic acid platform vaccines, the RNAs, have been shown to be 95% efficacious versus symptomatic disease, virtually 100% efficacious in terms of serious illness or death. They both are right now available under emergency use authorization. And probably if any of you have been vaccinated, it's been with one of these 2 vaccines. The Pfizer vaccine, actually, I think one of the production facilities is in Kalamazoo. The adenovirus vector, the J&J, Johnson & Johnson or Janssen, has been shown to be 72% efficacious in a study in the US, 85% efficacious overall in some other studies. As I'll show you in a moment, though, closer to 50% against the South African variant. It will be reviewed by an FDA advisory committee, I think, in the very near future, and it is anticipated that these data may be adequate for emergency use authorization. AstraZeneca is just presenting some of their phase 3 results outside the US, where they have efficacy ranging from 70% and up. Again, their efficacy against the South African variant is felt to be very low. This vaccine may be reviewed by FDA sometime late February or March. The protein vaccines are a bit further behind. They are in phase 2 or phase 3. We're hoping to see results from those studies again in the next couple of months, and then those should be out. At the moment, though, the only 2 vaccines out are the 2 RNA vaccines, and again, under emergency use. In terms of the time frame, one typically talks about years to develop a vaccine. This is the timeline on the SARS-CoV-2 vaccine, that the sequence was released by the Chinese January the 10th. And again, as I'm giving you the US view, similar things were going on in other countries, including China. Within a few days, that sequence was used to develop vaccine candidates through the RNA platforms. Phase 1 trials began within 2 months. Phase 2 within 4 months. Phase 3 2 months later, and then within a year, we had phase 3 efficacy data and emergency use authorization. It really is an incredible timeline. This shows you some of the data from the 2 RNA vaccines, the Pfizer vaccine on the left, the Moderna vaccine on the right. Again, we're looking at the incidence of new symptomatic infection. And this is an important point. A vaccine can prevent infection, it can prevent disease, or it can prevent death. Obviously, you'd like it to prevent infection, but even if it doesn't, if it prevents serious disease or death, you've got a winner. What we're looking at here is preventing any symptomatic disease. So the red line on the left is the placebo arm of Pfizer. The triangles are severe disease, and you can see while disease just crept up in the placebo arm, after about 10 days, there were very, very few new cases of symptomatic disease in the vaccine arm. Of note, there are examples of infections in the vaccine arm in individuals who did not have symptomatic disease. So again, even though you're vaccinated, you still can become infected. You still may develop symptoms. You may not, but you probably can still spread the virus. Hence the requirement that everyone continue to wear masking when they're in an environment where they might be able to transmit infection. The Moderna data, again, very similar to the Pfizer data. Placebo arm continuing to show infections, through 100 days post randomization. In the vaccine arm, after about 12 days, really no more infections. So if you're getting one of these RNA vaccines, while it's a prime and a boost, you really do begin to see some protection even before the boost. Just to talk a little bit more about the other candidates, the Johnson & Johnson human Ad26, again, 66% overall against moderate to severe disease. That efficacy was 72% in the US, but 57% in South Africa. Novavax, which is one of the protein based vaccines, announced their phase 3 results just a few weeks ago, claiming close to 90% effective against symptomatic COVID-19. About half of their cases had the UK variant. So again, good efficacy against the UK variant. The data they have from South Africa, though, suggests it's only about 50% effective against symptomatic infection. And again, here, most of the cases were that South African variant B.1.351. So again, this consistent theme that the South African variant may evade some of the current countermeasures. Just to mention that the US and the companies I've mentioned are not the only groups doing vaccines. The Chinese also, and the Russians also have very aggressive vaccine programs. The Russians looking at a recombinant human Ad26 or the recombinant human Ad5 boost, for which they've reported 90% efficacy. I don't have a slide on the Chinese vaccine. They're looking at recombinant Ad5 as well as inactivated viral particles. Just last, to make a few comments on vaccine distribution. These are the phases put forward by the CDC's Advisory Committee on Immunization Practices. This is a guidance that the states then take to decide how they want to distribute the vaccine that they're being provided by the federal government. Phase 1A being healthcare personnel, long-term care facility residents. The latter being the most susceptible to infection, the former being force multipliers, being able to maintain the healthcare infrastructure. Phase 1B, other essential workers, individuals over 75. 1C, individuals 65 to 74, 16 to 64 with high-risk conditions, essential workers that were not in 1B, and then phase 2, basically, for pretty much everyone else. Herd immunity is what we're looking for. That either happens after the virus has been through and infected most everyone who's susceptible in your population, or infected a segment and the rest of the segment has been vaccinated. One doesn't know how many people need to be immune to this virus to get herd immunity. The guesses are somewhere around 70 to 85%. Again, that stresses the importance of getting vaccinated, realizing that even if you're vaccinated, you still might spread infection. And really, keeping an eye out on these new variants. The other thing we don't know is how long immunity lasts, either after infection or after vaccination. We do know that after infection, you can become reinfected and you can become sick once reinfected. We also know that after vaccine, even 2 weeks after your booster, you can become infected. So far, we have not seen severe disease or fatal disease in individuals who've been vaccinated. This just gives you an idea of the rate of vaccination doses being administered in the US. So you can see we're getting upwards to about 1.5 million doses a day. And the final slide is this is just a wake-up call of how important it is to have infrastructures in place to be able to respond to emerging infectious diseases. The coronaviruses, obviously are a major threat from SARS to MERS to now SARS-CoV-2. There probably are ways to have universal vaccines that could hit this class of viruses. And a lot of the forward-thinking research right now is looking at ways to deal with classes of viruses as opposed to the single strain of a class of virus. So with that, thank everyone for their attention, and I'd be happy to take some questions.
Thank you, Dr. Lane. It's at this point that if we had a live audience, you'd be getting a standing ovation. I'm sort of a news junkie myself. And, I watch the news, I listen to the news, I read the news. But truthfully, I have not seen as comprehensive approach to the virus and the vaccine since this began over a year ago. And I'm very thankful that you are here today to share this valuable information with us. We've got time for some questions here, so let's go ahead and get started. What other vaccines have already been developed using either Pfizer or J&J techniques? I think you've probably already answered that one, but is there anything else you'd like to add to that?
No, the RNA vaccines have been studied actually for quite a few years, looking at respiratory syncytial virus. So there's an element of experience with these, but there are no licensed vaccines, certainly no widespread use as is currently in place. So we do anticipate there will be some rare adverse events that may be related to the vaccines that we'll see with the larger number that are being immunized. The AD26 and the Johnson & Johnson vaccine, that has been studied quite extensively for Ebola virus, and in fact is one of the WHO qualified vaccines for Ebola virus. There are no licensed AD26 platform vaccines in the US. Again, as I mentioned, the protein, that's pretty standard, tetanus, diphtheria, and others.
Here's another question. To what degree do you think the flattening of the curve messaging was helpful last spring and summer? That messaging seems to have disappeared in the fall, right when the largest spike was beginning to take place.
That's a really good question. I think one of the challenges, and I tried to illustrate it in talking about the response in China. I mean, you couldn't go anywhere in China. Keep in mind, this is at a time when they had very few new cases without, hearing about SARS-CoV-2, COVID-19, and what you could do to help the country fight against the virus. It was a war. The entire country was at war against the virus. I think one of our challenges was having a consistent message of that type that went out to the public. So flattening the curve, you can understand what that means. But what we really, the message I personally think we needed to get out was, you can prevent this virus from being spread. These are the things you need to do. Please do these things, if not for yourself, for those around you. And I'm not sure that everyone sort of grasped what the flattening of the curve, and you could see those three waves that I showed, whatever rhetoric we were using early on was not enough to get us to where we need to be. It really, I'm glad to see more public service announcements coming out now.
Good. Next question. Is it recommended for individuals who have the long haulers to receive the COVID vaccine?
So I'll generalize the question a little bit. It is recommended that people who have been infected still get the vaccine. It's been suggested that one wait about 90 days for that to happen. We don't know. It's interesting because not everyone who gets infected makes an immune response. So this is a little bit different than some of the other viral infections we talk about. So it is recommended that everyone get vaccinated. There certainly is speculation that part of the long hauler syndrome might be due to the immune response. So there might be some concern about immunization in that setting. I'm personally of the mind that until there's some reason for us not to do it, it's probably good to do it, because another hypothesis is that there may be some smoldering replication still ongoing. So getting a better immune response actually could be helpful for the long haulers.
Good, thank you. Here's one that goes way back to last spring. What is the status of the hydroxychloroquine therapy recommendation?
So hydroxychloroquine was one of, if not the first therapy that got emergency use authorization in the US. And that probably would be a whole separate lecture, if not series of lectures. There is a great coverage of that in Vanity Fair from several months ago that, for people who are interested, could look at. There was never any data supporting the use of hydroxychloroquine. There were people making claims about it with small, what we would call underpowered studies. When people did the right type of research, the randomized placebo-controlled trials, hydroxychloroquine was shown to have no benefit, and its emergency use authorization was actually revoked.
Thank you. Next question. Will people who are vaccinated test positive with an antibody test? And if so, for how long?
So yes, if you're vaccinated, in all likelihood, you will develop antibodies to the spike protein. So if your antibody test measures antibodies to the spike protein, you would test positive with an antibody test, certainly not with the antigen or the RNA test. There are tests for antibodies to other parts of the virus. So you can do a test for antibodies to the core protein or the nucleoprotein of the virus to distinguish a vaccine antibody response from an infection antibody response. We don't know how long the antibody responses are going to last, either with a natural infection or a vaccine. That's part of ongoing research.
Next question. Now that we've had our first adult diagnosed with MISA, what treatments are going to be initiated to recognize prior to multiple system failure and death?
Yeah, I mean, fortunately, MISA is a very rare condition. The approach is similar to the approach in children, intravenous immunoglobulin and corticosteroids. Again, that can occur a time after infection, and it's very likely it's somehow related to an overactive immune response. Given its low frequency, there are no recommendations now to go further out with immunosuppressive therapy. Really, the dexamethasone for 10 days is the standard therapy for severe disease. If we learn more, then we may need to think about that differently. The key, I think, is being able to identify who may be at increased risk for it and see if we can't do something preemptively in that subset of patients.
And here's a question I know that many people have discussed over coffee and socially when they have an opportunity to meet, probably over Zoom. Here it is. We are social beings. Masks and social distancing has affected the mental health of many individuals. I've missed hugging and visiting with my grown children. At what point in the vaccination process will we be able to be with family and friends for holidays and important events like graduations and weddings without being fearful of spreading or catching COVID?
Yeah, that's such a good question. I would say that is basically the question of the day. And while everyone wants to give an answer, there is what I say is there's a light at the end of the tunnel. I just don't know how long that part of the tunnel is right now. I do think if the projections on getting vaccine out are correct and we get, most everyone vaccinated, let's say by the midsummer, I would think going into the fall, we should be back to that setting where we can, interact, much more like we had in the past. If we have the ability to do our own diagnosis, I mean, it's very frustrating for everybody. I know if, you could do a simple test at home that was inexpensive. So if you had people over before we really had herd immunity or really had this infection under control, you could test on the spot and then have some assurance. I mean, I can tell you what I do. So I've been vaccinated, but I get tested here at our hospital once a week. And it's only if I've had a negative test and I know I'm vaccinated, would I even think about being in a setting with another person that's not part of my family, my immediate family, without wearing a mask. So I think now is the time for us to really be as conservative as we can, because you see that curve going down like this. Whether it's a variant or not, that curve will go down if we do the standard public health measures. So now's the time, sort of like to me, the third, lap of a 4-lap race or something like that, to not let up, because we do have things moving really in the right direction.
Thank you. Do you have a response to people who believe the vaccine and mitigation strategies aren't necessary because the death rate is so low?
So let's say the death rate is 1%. So that sounds like a low number until you apply it to a population of 300 million, which would be 3 million people in the US. And I would be quite surprised if the families of those 3 million, which probably give you another, 15, 20 million, would feel that anything that we can do to prevent death of this virus is something that we should be doing.
This one sort of follows along with that last question, and I know it's a question probably without a real clear-cut answer, but here it is. what should we or could we have done differently to have controlled COVID-19?
There will be multiple, multiple sort of after action studies. There will be, in quote, lessons learned. There will be independent bodies charged with looking at this. And I'm sure there will be a variety of things. But the one thing I can say is not that I feel we should have a society like China's where everyone, does exactly what they're told to do or there's serious consequences. And the close immediate contacts in China were actually pulled out of their families and put into quarantine camps. So that's not something that we would aspire to. But I think having a consistent message from the very top leadership all the way down to the local leadership, a consistent, simple message. This is a serious problem. People are going to die, but we can prevent a lot of this if we do the following. And just if we had just reinforced that, if we had public service announcements, I can't recall seeing public service announcements from high profile individuals, encouraging the American public to do what we know can get this under control. I think that's what I really wish we had been able to do better.
Question about what lessons has the NIH learned from this pandemic?
So one thing that's interesting, we just focus it to NIH. One of the things that we, think we should be able to do better is to rapidly launch large clinical trials with the right design. One of the things that happened was a lot of very small studies got launched in a hurry, giving conflicting data. Hydroxychloroquine is a good example of that. We need to be able to launch big studies in a hurry. They did that in the United Kingdom. That's where the dexamethasone result came from. So we need to, I think, have within our clinical research infrastructure the ability to rapidly launch large, well-designed trials so we can answer the critical questions quickly.
Will the variants affect the level that we need to achieve herd immunity?
It's a good question. It doesn't probably change the level of people who need to be immune to the variant, but it may change, the level of immunity that a person has. So when you talk about 70%, 75%, 85%, you're talking about 85% of the population, being close to 100% immune. So if that population is not 100% immune, but only 50% immune, then you don't have herd immunity. So in fact, it's quite possible that the variants will affect the time it will take us to get to herd immunity.
Dr. Lane, what's your view on the possibility of a vaccine being needed annually, similar, for example, to a flu vaccine that we take now?
It's interesting because we don't have a lot of long-term data on the RNA vaccines. We don't know how rapidly the antibody response or the protection may fall. Antibodies are one measurement of immunity, but they're only one measurement. The immune system, had two main elements triggered by the vaccines: B cells that make antibodies and T cells that kill infected cells. So both of these elements are brought into play with the vaccines. How long that allows someone to stay protected, we really don't know.
We're coming right up on the 3 o'clock hour. Do you have time for maybe a couple more questions?
Sure, I'm happy to.
All right.
I do think, as you were asking earlier, though, just to complete the answer, I do think as we learn the duration of protection, as we see infections in the vaccinated individuals, we'll get a better handle on the duration of protective immunity, and with that, probably design strategies that I'm guessing would lead to the need for booster immunization periodically, and potentially booster immunization with slightly different vaccines that have accommodated the changes brought in by the variants of concern.
Here's a question that I know comes from a corporate person. Most corporations have pandemic plans in place, in the event their employees can't come to work, especially customer-facing organizations. Was there a pandemic playbook in the federal government, in any aspect of the federal government?
Yes, there was. And the question to follow up that I won't avoid answering is, was it applied? No, it wasn't.
Was there a reason for that?
I think it's politics to a degree. A new administration doesn't always value the work of the prior administration. You know, really on the heels of the Ebola outbreak during the Obama administration, a lot of work went into pandemic preparedness, a lot of work, you know, both from the public health measures, the research measures. There was a playbook.
Thank you. Flu. The flu, your chart indicated that the flu this season seemed to be way down. To what do you attribute that?
It's a really good question. Everywhere you look, so we could start off looking in the Southern Hemisphere during our summer, flu rates are like the lowest anyone has ever seen them. And I think that's an absolute reflection of the social distancing. I think what prevents us from transmitting SARS-CoV-2 prevents us from transmitting influenza. And the irony is there has been a lot less illness from influenza, and it makes you think, you know, well, maybe, you know, there's times where the masking might be used a little bit more.
Dr. Lane, is there a resource for people with comorbidities for them to access information about what to do, how to protect themselves, whether they should get the vaccine? Is there a place for them to go to learn more about this?
Yeah, I don't know. It's not that I've got one good website that I could tell you. I think really the more you can put into search terms and then see what you get. I mean, I work for the US government. I tend to rely on the .gov websites a bit. If there's something at cdc.gov, I think that's usually pretty good information. There is a federal-wide COVID-19. There's an HHS-wide COVID-19. Many of the professional societies are also putting out guidance for the diseases that they are specialists in.
Good. I thank you for spending a little extra time with us to take some of these questions. Dr. Lane, there were close to 200 people online today listening to this. And in terms of the speaker series, that's a very, very good turnout. And what's interesting is they stuck with it. And so it's a huge compliment to you. We've always sort of gauged the success of our speakers by the number of questions that come in. And you have set a record there, definitely. So that is excellent. And I just want to say to you, on behalf of everyone attending, on behalf of the board of directors and our sponsors, thank you so much for spending the time with us today.